Introduction. As we age, eye cells gradually lose energy and function, partly because their mitochondria, the cell's batteries, become weaker. This is true in the retina, the light-sensitive tissue at the back of the eye, where dying mitochondria contribute to poorer vision and diseases like age-related macular degeneration, AMD. Photobiomodulation, PBM, is a gentle therapy that uses low-intensity red or near-infrared light, usually around 670 nanometers wavelength, to stimulate cells. Laboratory studies suggest that shining 670 nanometers light into the eye can recharge mitochondrial function, boosting energy, ATP production, and easing inflammation. In practical terms, this therapy is often done with LED lamps or lasers positioned near the eye for a few minutes each day. Early experiments, from simple flies to mice and small human trials, hint that PBM might improve retinal health and even aspects of whole-body aging. This article reviews how 6 and 70 nanometer light benefits photoreceptors and retinal ganglion cells, summarizes results in experimental models, including lifespan effects in insects, and discusses dosing, safety, and possible home use. Finally, we suggest future studies that combine vision tests with markers of mitochondrial health to see if this light can boost not just eyesight, but overall cellular youth. How near-infrared light boosts retinal cells. Photobiomodulation at 670 nanometers targets mitochondria, the tiny structures inside cells that make most of our energy, ATP. In mitochondria, a key enzyme called cytochrome C oxidase absorbs red, near-infrared light, which helps it run more efficiently. In effect, 670 nanometers light raises the electrical membrane potential of mitochondria and lets them crank out more ATP. Studies show this extra energy relieves age-related decline. For example, one report found that a month of daily 670 nanometers light in old mice roughly corrected their low mitochondrial membrane potential and ATP levels. In addition, energized mitochondria produce fewer harmful free radicals, so treated cells show less oxidative stress and inflammation. Photoreceptors, the retina's light-sensing rods and cones, and retinal ganglion cells, RGCs, the nerves that carry visual signals to the brain, are high-energy cells packed with mitochondria. By boosting mitochondrial activity, 670 nanometers light helps these cells work more efficiently. Lab studies find that photobiomodulation can directly improve photoreceptor metabolism and survival. For instance, in a mouse model of light-induced retinal damage, 670 nanometers treatment greatly improved photoreceptor health. Treated cells had stronger mitochondrial respiration and less stress-induced damage. Likewise, in an optic nerve injury model, 670 nanometers light preserved RGCs. Treated rats showed a 3.4-fold increase in visual signal strength and 1.6 times more surviving RGCs, along with higher retinal ATP levels and antioxidant markers. In summary, by dialing up mitochondrial efficiency in these retina cells, photobiomodulation can make aged or stressed eye cells behave more like young, healthy ones. Results from animal studies. Researchers have tested 670 nanometer PBM in various aging and disease models with encouraging results. In aged mice without injury, daily 670 nanometers exposure for 1 month led to markedly better retinal function. Electroretinogram (ERG) tests showed roughly 25% higher rod and cone responses in treated old mice, approaching the levels of young adults. In other words, older mice had significantly stronger vision signals after 670 nanometers treatment. These improvements likely stem from giving photoreceptors more ATP to fuel their light detection ion pumps and from quelling low-level inflammation in the aging eye. In models of retinal degeneration, like inherited macular degeneration or diabetic damage, PBM also helps. For example, red, near-infrared light reduced harmful immune signaling from supporting Muller cells and protected photoreceptors from degeneration. In a mouse model of oxygen-induced retinal injury, pre-treatment with 670 nanometers light protected photoreceptors and inner retina, reducing cell death and preserving function through similar mitochondria-based mechanisms. These studies show a common theme. 670 nanometer PBM quieted stress signals and boosted cell survival in aged or diseased retinas by enhancing mitochondrial energy. Fruit flies and longevity. Remarkably, the benefits of 670 nanometer light extend beyond vision and even beyond mammals. A striking example comes from fruit flies, Drosophila. Because flies age fast, researchers exposed them to 670 nanometers light each day and then measured their ATP, inflammation levels, mobility, and lifespan. Early on, treated older flies had about 80% more ATP and 15% less of an inflammation marker in their bodies. More importantly, many more 670 nanometers treated flies survived into what is considered old age. At peak difference, treated flies outnumbered controls by over 100%. In other words, far more flies lived into the later part of their life, even though the absolute maximum lifespan, when all eventually died, was similar. Treated flies also climbed higher and moved twice as far as controls at 7 weeks old, showing better mobility. In brief, daily red light nearly doubled the fraction of flies making it to old age and kept them more active. These dramatic findings suggest that photobiomodulation can act on whole body metabolism and health, at least in insects. Evidence in small mammals. Direct lifespan experiments are harder to do in mammals. Their long lifetime and light penetration limits make it tricky. However, numerous rodent studies hint at general health benefits. For example, treated old rodents show lower tissue inflammation and higher ATP in the brain and retina, similar to the photoreceptor data above. Some studies in aging mice have found that regular near-infrared exposure improves muscle function or cognitive test scores, likely by the same mitochondrial boost. While not as dramatic as in flies, these studies support health span improvements, better function in old age. For instance, in a rat eye injury study, 670 nanometers light in the days after damage dramatically cut oxidative stress and increased protective factors like NRF2 and PGC1-Ha involved in mitochondrial growth. Overall, animal work suggests that even in mammals, PBM can power up aging tissues systemically, though true lifespan extension is still under study. Early human feasibility studies. Given the promising lab results, small human trials have explored PBM for aging eyes. One pilot treated 31 older adults, some with early AMD, some with normal retina, using a handheld 670 nanometer LED held close to the eye for 2 minutes each morning. Over a year, these patients were tested for visual acuity, low light vision, and retinal scans. In the healthy, no AMD group, there was a small improvement in dark-adapted scotopic vision thresholds by 1.8 dBs, indicating slightly better night vision after a few weeks. However, in eyes that already had intermediate AMD changes, there was no significant change in vision or retinal structure at any time point up to 12 months. In other words, once AMD is established, 670 nanometers for a year did not reverse it in that small study. These mixed results echo an earlier anecdotal report. One non-controlled case series of 18 AMD patients claimed reduced drusen deposits and some visual improvement after 12 months of 670 nanometers light, but this was not confirmed in larger trials. Importantly, all of these studies found the treatment to be safe and well tolerated. No serious eye damage was reported, and a few participants only withdrew due to the burden of testing rather than any harm from the light. Transient mild glare or sensitivity is the only complaint ever noted, and it is rare. Thus, early human work suggests that 670 PBM is easy and safe to apply, and it may slightly help normal aging eyes, e.g. night vision, but it needs more study to prove any benefit in eye disease. Dosing and safety. Research studies have used modest light doses. For example, aged mice were typically treated with 40 milliwatts per centimeter squared for 15 minutes daily, about 36 Gu per centimeter total per day. The human pilot described above used 40 milliwatts for 2 minutes, 4.8 Gs per centimeter each session. In general, PBM uses very low power lamps, far less intense than the sun, so there is no heating or burning. Sessions range from a minute or two to about 15 minutes once or twice a day. Many studies repeat this daily for weeks. Because 670 nanometers is outside the damaging blue UV range and is used at lower radiance, no harmful effects on the retina have been seen in trials. By contrast, too strong light can harm eyes, but PBM devices are specifically calibrated to safe levels. Home-use devices are already available for skin and even eye PBM. These often use 670 nanometer LEDs at known safe power, usually tens of milliwatts per centimeter square. To apply to the eye, a patient simply needs to hold or place the light a few centimeters from the closed or open eye for a few minutes. Because no special alignment or dilation is needed, a session feels like a warm red glow. Some studies gave the light with the pupil open, while others even treated through closed lids. In either case, 670 nanometers penetrates enough to reach the retina. As a safety note, patients should never look directly into very bright lights, but typical PBM devices are designed to avoid glare beyond mild brightness. Overall, nightly or daily sessions at home are practical, much like using a small light panel for a few minutes to improve sleep or skin health. Practical considerations and future studies. While preliminary, the evidence encourages well-designed trials of 670 nanometer PBM in aging eyes. Future studies should combine traditional vision tests like acuity charts, dark-adapted threshold tests, contrast sensitivity, or retinal imaging with measures of mitochondrial health. For example, one could track blood markers of metabolism or inflammation, such as lactate, ATP-related factors, or antioxidant levels, before and after PBM therapy. This would tell us if the light's energy boost in the retina is accompanied by changes in the rest of the body. Combining ophthalmic outcomes with systemic biomarkers would clarify whether PBM is simply a local eye treatment or part of a broader rejuvenation effect. Trials could also vary the dose, duration, and power to find the optimal recipe for patients. Moreover, long-term follow-up would check if PBM slows progression of diseases like AMD. Overall, 670 nanometer photobiomodulation is a non-invasive, low-cost approach that may help aging eyes by restoring cellular energy. It has now shown benefits from insects up to mammals in lab studies, and has shown some safety and feasibility in humans. By rigorously testing it in future clinical trials that measure both visual function and mitochondrial activity, researchers can determine if shining a little red light every day might keep our retinas, and perhaps the rest of us, healthier for longer. Conclusion: photobiomodulation with 670 nanometer red light appears to revitalize aging retinal cells by heating up their mitochondria. In animal models, it boosted vision signals, preserved photoreceptors and RGCs, and even extended healthy lifespan in flies. Early human trials show it is easy to use and very safe, though the benefit in eye diseases remains to be proven. With proper dosing, few minutes daily of low-power LEDs, and eye protection guidelines, home-based 670 nanometer therapy is technically practical. The next step is rigorous patient studies that jointly assess eyesight and cellular energy markers to see whether this gentle light can light up not only our eyes, but our overall health as we age. All links to sources are available in the text version of this article. You can find the full article at visualfieldtest.com. Thanks for listening. To check your visual field, click the link at the bottom of this article or visit visualfieldtest.com.